So, good evening, everyone, and welcome to the webinar on eDNA barcoding. And I thank Barcode Biotechnology for inviting me for this wonderful webinar. So, who is this Barcode Biotechnology? They are my sponsors. They have to introduce them. And they are founded in 2013, and it's basically a DNA sequencing service provider company. Their infrastructure consists of Sanger sequences, Illumina platforms, and also they have Mermaid system for synthesizing primers, and they have a PCR platforms for providing consistent and high quality DNA sequencing services. And these are their some of their services. They are providing mixed culture identification service. They are providing RKL identification service. They are using 6NS rRNA for prokaryotic species identification, and they are using ITS for mushrooms. They are also doing multi-locus gene sequencing in case you want precise identification, especially for bacteria. Multiple molecular taxonomic genes can be targeted, and similar case for phages also. So for insect barcoding, they are using C1 sequences for animals. C1 sequences are used for identifying various animal phyla. And they are also doing meta barcoding. You can even send a community of samples. They can decode all the species in your group of samples using CO1 gene sequencing. I can challenge they are most affordable DNA sequence service provider company. And they are also doing wildlife forensics, where if you can send them a fecal samples, they not only tell you the host or animal species the fecal belongs to, or they can also decode the entire dietary composition of the fecal. What the animal have fed, they can be done through metabarcoding. Even for birds, most of the birds are restricted from sampling because of their IUCN status. You can simply scrap off the fecal material and decode the bird species from that. Even for plants, not only fresh tissues of the plants, even if you send them a preserved powder or herbal powders, we can decode what species of plant is this. So even if you do not provide them any sample, just a air filter through a filter membrane, we can decode the entire biodiversity from the air. So our this webinar is all about decoding environmental DNA, where we can sample water, land, soil, and air samples. So that's the services. You can follow this. I hope many of you have followed. That's why you are listening to this today. So follow this channel. And also, apart from sequencing services, they also provide Barcode Biotech provide internships. Okay, this is something very unique internship in India. Actually, it's an online internship, bioinformatics internship, where the clients are actually interns are trained in handling sequencing files. But you can also send your samples for barcoding or sequencing. In case of this internship to be started by July 16th, you can send them a microbial sample. Two samples are sequenced for free. Using your own machine output file, you can analyze your sequences, you can do the blast analysis, phylogeny, pairwise distance data. You can build an entire manuscript around it. They will help you in creating a manuscript writing also. So this is what this internship is about. UG, PG students can take it. You can provide them a pure cultures of bacteria, two cultures or one pure culture, and ask them to go for multi-locus gene sequencing, where 6NS rRNA and other RPOB genes can be targeted. You can even send them a mixed culture and point out the bacterial and fungal colonies in the plate. They will do the pure culturing. They will sequence it, identify it, and they will send back the glycerol stock for free. Okay, these are all packages that is covered in the internship. Even you can send them an environmental sample, soil, water, any milk samples. They can isolate microorganism. They will show the mother picture to you and ask what are the colonies you need. They will sequence it out of free. If you are intern registered under this program, which is going to start by July 16th. You can send them a plant leaf animal samples and get them two sequences in a species or two different plant species. You can sequence for free. So gut sample, they will give you the CFU count, and also they will sequence two bacteria for free. So this is an internship fee they have. Anyone who is doing UG, PG, if you are especially third year of UG, you can have a head start. If you are exploring some microorganism for bioprospecting aspects, you can identify them using this internship that is going to happen for 21 days. You will be trained in various aspects with your own data that you are sending to the company for sequencing. Okay, if you're a PG student, still it's a what's good service because you can focus on application part now during your third semester and fourth semester. Many of you might be aspiring to go for European countries for for further studies. Having a publication is important. So it's having a quick molecular data in most affordable price and writing a manuscript around this will be an ideal topic here. And also, if you're a PhD scholar, you can frame one separate unit in the chapter as a molecular taxonomy. Even within 21 days, you can get your own data and you can learn how to analyze the data. The main reason behind this online internship is for many of the clients Barcode Biotechnologies are handling, they are actually coming as a sequencing customer. Then they ask for sequence analysis. Then they ask for discussion part. Many times we end up writing the manuscript for them, and many times we are writing a proposal for faculties who can also get the if you're a faculty, you can create your own team in DNA sequencing and barcoding in your college or university. It's very much possible under this program. And even non-microbiologists who are environmental science, agriculture people, the veterinary microbiologists who want to study microorganisms for their animal, the plant, endophytes, epiphytes, botany guys, they can also make use of this fellowship. So this is open for everyone. Those who can register, they can avail these services, two sequencing services free under this fellowship. So apart from giving a DNA data, what this internship will cover is you will learn the machine output file for your sequences, how to analyze the machine out DNA chromatograms, how to classify the peaks, how to extract the sequences, what are the sequences we need to prepare for further analysis, how to compile forward and reverse sequence using bioedit programs, and how to use the online tools like BLAST to find what species it is, and how to prepare a multiple sequence file, alignment file using ClusterX programs. These are all free softwares, molecular revolutionary genetic analysis, mega software, which is being used for constructing phylogenetic tree, how to prepare a Kimura two-parameter pairwise distance data. And if you want to work as a bioinformatic research associate in many of the sequencing companies, you should know how to prepare a sequence identification report, a species identification report. So all these trainings are done, and how to submit the sequences in NCBI, GenBank, or even India's own database, INDA, and how to get the accession number, how to define a barcode gap, how to use your own sequences and the reference sequences from NCBI to define barcode gaps. Even if you end up sequencing yours, if no one has sequenced that particular gene in your organism, you may end up having a first time barcode data which can be easily published. This is a proof. This is one of my paper. I am not a variant taxonomist. I am not a zoologist, but I worked with the zoology. When I do the blast analysis, actually done from Barcode Biotechnology, they told me that it is a first time barcoded species. So I named the paper something like this, and we got it published. Even here also, even if it is repeatedly barcoded species, but Barcode Biotechnology will train you in defining the barcode gap, which can still be published like this. So we have marine fungal barcoding analysis, and where we have proposed, this is one of my paper, I have proposed the barcoding gap for first time and got AC accepted. So if you are someone who is aspiring to have a quick manuscript in DNA barcoding, I think this internship is a gateway. So I recommend do use it. So coming to today's topic, eDNA barcoding. Why do we need this? Before we proceed, let's see what is barcoding, what is DNA barcoding, and what is meta barcoding. Then it's evolved form, that is environmental DNA barcoding, eDNA barcoding. Okay, so let's talk about biodiversity first. Then we will understand why barcoding is essential. The biodiversity, we are actually in crisis period. Okay, we are 2.8 billion people as of 2 p.m. of June 26th data, just last week data. Our carrying capacity is actually 3.3 billion. Anything over that, we have to sacrifice various biodiversity for perpetuation of Homo sapiens. That's what's been happening. We reached 3.3 billion to the to whatever land area we available. We reached that population by 1965. Okay, so now we are at least three times over the actual biodiversity the Earth can hold. So biodiversity crisis is true. We have lost almost 68% of diversity when compared to 1970s diversity. And the value we are paying for the loss is more than $44 trillion. Okay, it's very clear that human population is a main factor driving the biodiversity loss. So if you take Japan, where their human population is reducing, so if you ask the question, can we repopulate the diversity if human population is going down? The answer is no. This paper came 30 days back. You see, biodiversity change under human depopulation. And Japan is an ideal area to study this depopulation. So what they found is even if you change the landscape, or when the landscape is changed permanently to accommodate growing population, biodiversity loss is permanent and it cannot be reversed. That's a crisis they recently discovered this month. So what is this DNA barcoding? Every living organism have their own DNA fragment or DNA barcode. Okay. Those DNA sequences are unique and the sequence can be used to identify the species name. In other way of saying, if there is a taxonomist who can identify a particular species, if I can make a DNA sequence, a barcode gene, if I can sequence it and put it in a database, now a non-taxonomist who want to identify that particular animal just have to sequence that gene and compare the sequence with the database. Already I have identified a particular species, I have deposited the sequence. So when I just compare it with the reference database, boom, you got the species name. That's how barcoding works. To simplify that, imagine you have a Word doc file writing all your friends' name. Each and every friend has a unique DNA sequence. When you press Control Find and find a particular input a particular sequence, whichever sequence is getting matched, that friend's name will be displayed as a match. So that's how barcoding works. We create a database with known species and their barcode gene, and we will use those as a reference database for any non-taxonomist or even general public who want to identify a particular species. Now can send the sample to a sequencing company, and the sequencing company will sequence it, and their bioinformatics associate will do the blast analysis and give you a species identification report. So that's the simplest explanation of barcoding. So what is this? Why we call them as a barcoding? See, if you go to a supermarket, you have a various product with UPC, Universal Product Code. If you scan it, the entire history of the product is revealed through the scan, right? Likewise, scientists came up with this idea. A particular barcode gene inside all the organism can be read and can be matched with their species name, which can be used as a reference sequences to identify the species as biological system to belong to a particular species. Okay, so can why we need such a technology? You see, last 200 years of taxonomic science have documented only 1.5 million species, where we are, in my estimate, I have seen there are at least 10 million species in this biosphere. Okay, so it's very clear that thousands of species may have evolved and gone extinct without even we document it. Okay, we are losing biodiversity in this lack of technology to speed up the documentation. That's when barcoding technology has come into picture, where this is not something very new. Okay, in microbiology, people know for last 40 years we are using 6S RNA sequence to identify bacteria. Even in animal molecular taxonomy, we were using various genes from different phyla to identify various organisms. The only problem in using different genes is you cannot do a comparison study. If I use 18S rRNA to identify fish and ITS region to identify the polychaetes, marine worms, we don't have a common gene base to compare it and we can identify it. So that's when DNA barcoding becomes very handy. So all the thing has started with a simple idea that why not we have a same gene to document entire life. So it started with mitochondrial DNA, cytochrome C oxidase subunit 1, CO1 gene, okay, where scientists identify the known species, sequence it, put it in the database, they can compare it and they will know the species. But this CO1 gene is lacking in the plant system. Okay, they are very much conserved. There is no variation among CO1. They are not present in bacteria, or if even if it is present in some yeast species, they have there is no much conservation within the species. Okay, so right now these are the four barcode genes, I would say, excluding prokaryotes, that is archaea and bacteria. These are the four barcode genes: CO1 gene for animals, RBCL for plants, and matK. The plants have two genes, angiosperms and gymnosperms, and fungal ITS gene is working. So this is the how the entire barcoding can be explained in one picture, where if you want to identify a species, extract the DNA, go for sequencing. Those sequences can be matched with the database, and boom, you know your species name. Okay, so everything started with this professor, Paul D. Hebert. He's still very active in service. He's from University of Guelph, Canada. He came up with the idea to use this single gene. He is basically an entomologist. He proposed this idea. After this, you can see in every country it is picking up. Polish barcode of life, UK barcode of life, Bulgarian barcode of life, even India. Yeah, USRSA and Botanical Survey of India, they did a good job in initiating this. Even phylum specific consortia are present, like Lepidopteran barcode, sponge barcode, plant working groups, fish barcode of life. So everyone is because you have seen we need to have a perfect database to facilitate this technology. All known species should be identified first, sequenced, and deposited in the database. That's how we can actually, have a Pakka database so that barcoding works. Okay, so everything started with this biological identification through DNA barcodes during 2003. After 6 years, during 2009, they realized that the C1 gene is not working for plants. So they have analyzed all these genes and they proposed RPOB. Sorry, they proposed RBCL gene and matK gene for plants, and ITS gene has been proposed for fungi. Even here is a paper. When this paper came out, we tried to evaluate the ITS gene in marine fungi, exclusively yeast and molds. We saw that this gene is working Pakka in clearly delimiting all other fungal species. Okay, so in local scenario in India, we have, I would say, Center of Advanced Study in Marine Biology is a pioneer in DNA barcoding because they got a project from Census of Marine Life USA during 2008, and the first research article is published during 2009 in Current Science, which is India's first research article in DNA barcoding. And thereafter, National Bureau of Fish Genetic Resources is doing very good job, and other institute have simply picked up the momentum. But I would still say the efforts are not that up to mark. So here there are various other groups who are working on various consortia of barcode of life. So like I said, we have National Center for Biotechnological Information from USA, accumulating most of the DNA data. DDBJ EMBL is also doing good job. Matching to them in India, we have initiated this initiative 10 years back. They are slowly building up the data in silence and they are actually doing a very good job. We also have a dedicated database for barcode of life, which accept only those 4 genes. C1 gene for animals, RBCL for plants, matK for plants, and ITS for fungi. So only these 4 genes can be submitted here. If you want to identify, for example, a fungal species, just go for ITS gene sequencing, go for whole database. You can paste your sequences and know the species here. Okay. So this is how the barcoding works. Once you deposit the sequences in the database, say for example, this is a reference sequence in the database. Once you sequence your own gene, you can match the gene. Every nucleotide is matched with the database and you can identify your species. Okay. So that's how the barcoding works. It's very easily said, but once you want to barcode a species, you need to collect it. All key morphological features should be very carefully documented in multiple sets and then go for sequencing. Those sequencing along with the metadata should be submitted to the database. And after submitting the data to the database, now they are available in the database as a reference sequences, even a non-taxonomist, anytime, anywhere, in any place. Just sequence that gene and compare it to the database, you will know what species it is. So saying that's how the barcoding works, but it's actually recurring. If you want to barcode, you require a multiple skill sets. Okay, you need to be a taxonomist, conventional in particular area. You need to know how to do a DNA extraction and PCR as a molecular biologist. When you have a chromatograms, you need to know how to be a bioinformatician in analyzing the chromatogram and extracting the sequence and submitting the sequence to various databases and getting the accession number and creating you need to be a data analyst to maintain a barcode database. So basically this internship what our barcode bioinformatics is doing, that actually starts from here, where you will get your own DNA data out of samples you are submitting to the company. They will provide you this data and they will teach you how to analyze the data and maintain the databases. Okay. So speaking about this, let's spend few minutes in understanding what is this DNA barcode gene before we proceed for eDNA barcoding. Okay. Can an organism will have thousands of genes? Can everyone act as a barcode? The answer is no. For example, if I analyze the amylase producing gene between bacteria, between different phyla of bacteria, maybe there is not even single nucleotide variation. When I compare the nucleotides, they are all exactly same. But when I analyze the 6NS rRNA, every species have variation in certain segments. When I say certain segments, 6NS rRNA is 1500 base pair in length. We say base pair, these are nucleotide bases. Actually, they are complementing each other, so they are occurring as a double stranded DNA. Whenever I say base pair, so this 6NS RNA for E. coli is completely different from 6NS RNA of Salmonella or any other bacteria. Okay, that's why they are acting as a barcode gene. So other way of putting this question is, what make a gene to act as a barcode? Okay, so a gene should be having low variation within the species and high variation between the species. When I say between the species, there are multiple species under genera, multiple genera under family, multiple families under order. That's how the hierarchy taxonomy works. Okay. So the variation within the species should be relatively low when compared to the variation between the species among same genera. So what is this relatively low means what? So here is when the barcode, the vocabulary of barcode gap comes in. For example, in bacteria, the barcode gap is 3%. Okay, meaning if I want to call a sequence belonging to Escherichia coli, E. coli, they need to share at least 97 or more percentage of similarity with the reference sequence. When I say reference sequence, the sequences of E. coli in the database. Okay, I can have a similarity of 97 or 98, 99, 100, anything more than 97. If I have a 96% similarity, then my sequence is Escherichia, but not a coli. Okay, that is called species cutoff because the species cutoff is 3%, meaning 97% similarity or 3% variation. Okay, anything within 3% is a species cutoff for bacteria. I know for certain species there is a specific cutoff. For example, Salmonella will not come under this 3% cutoff condition. So those things an exception. So any gene that is having relatively low sequence similarity, low variation among the species and a high variation among between the species, we can use those gene as a barcode gene. That's how many genes have been experimented, and in plants they have announced cytochrome C oxidase 1 as a barcode. So when I say variation, how they are comparing? So imagine there is a one strand of DNA here, another strand here. Every nucleotide basis is matched here. Okay. So say for example, when you have a 10 nucleotide base pair, okay, only one nucleotide is varying among those two strands, then I could say there is 10% of variation. In 10 nucleotide, one nucleotide is variable, so 10%. Or in 100 base pair length, 10 nucleotides are variable, then I can call it as a 10% variation in 100 nucleotide base pair. Okay, so when I say species cutoff is 3% in 100 base pair, at least 3 nucleotides should be varying, is allowed to vary within the species. If 4 nucleotide or 5 nucleotide is variable, then that could be different species within same genera. Likewise, we have genera cutoff, which is around 6 or 5% in bacteria. We have family cutoff, which is around 7 to 8% in bacteria. We have order cutoff, class cutoff. Likewise, in every hierarchical taxon group, there are different cutoffs which are manually created. We will see how to calculate those cutoffs. Okay. So, let's see a little more deep about what is this animal barcode. Cytochrome C oxidase is a mitochondrial DNA gene which is actually 1500 to 1600 base pair length, but first 650 base pair in this cytochrome C oxidase is considered as a DNA barcode gene. Okay, not this one condition of having low similarity or no low variation within the species and high variation between the species is that's one condition. That gene should be in multiple copies. For example, here mitochondrial DNA is selected because they are circular, more resistant to degradation when compared to genomic DNA. So preservation will not be much problem. They are occurring in multiple copies within mitochondria. There are multiple mitochondria in within a single cell. So the copy number is high. So facilitating for PCR amplification. So all these characters are essential. So similar characters are exuded in chloroplast genes also, like maturase K and RBCL. Okay, that is used for barcodes of plants and angiosperms and genopsperms. Now here we have ITS region. Those are called as genes, RBCL gene, matK gene, CO1 gene. We call this as an ITS region because it is a combination of 5 genes. Usually it starts from the flank region of 18S rRNA subunit. Then they include ITS1 gene, ITS2 gene. In between, they have a 5.8S gene. And then it ends with the initial fragment of 28S large subunit rRNA. This entire fragment length is from 500 to 650 base pair, which is considered as a potential barcode, internal transcribed space region for fungi. When I say fungi, it works for both molds and yeast. For bacteria, we have 16S RNA. There are multiple variable region, which is used for identifying both bacteria and archaea. Okay, so barcode gap. You even if you are registering for this internship, you will be learning about how to calculate this chimera two parameter genetic distance using Mega software. In the Mega software, they will calculate within the species variation, between species variation, and they will do this numerical calculation. Okay, so for many domain in zoology and botany, I would say barcode gaps are not defined. If you can, sequence one species and download the reference sequences and define a barcode gap, that is some new data you can contribute in the manuscript, which most of the reviewers and editors are looking to publish a paper. Okay, defining barcode gaps specific to species, genera, families, whichever domain you are working. Okay, so what are the applications of DNA barcoding? Because we can create a library now, even anything, anywhere anyone can actually identify the species name. Since we have a common gene CO1, even I can compare the phylogenetics between the fish and a human. If I can go for the CO1 gene sequencing, there are many canned food, tuna food, and many canned foods. So that you never know what is the species advertised in the can is actual species preserved inside the can. Simply take a piece and go for barcoding. Now you know the species name. Even many red listed threatened species, when caught in the net by fishermen, these days they used to cut it out, chop it out into meat, and they bring it on ice because officers cannot recognize what species it is. But using barcoding now, just 100 milligram of tissue is enough to say what species it is. Okay, so likewise, they have multiple applications in detecting the hidden diversity. Even when you go for aquatic diversity sampling, you will know manual sampling for diversity assessment usually tend to miss many things. Okay, so they can precisely use for biodiversity monitoring, phylogenetic studies, forensics, conservation management. I still remember in southern India, there are many people still catching ornamental fishes from coastal ecosystems and they are selling it. Many officers had a difficulty in identifying the species, is it red listed or threatened, but using barcoding now precisely we can identify. Okay, and many forest officials are still using this technology to solve various mystery and cases. Okay, even adulteration in the industry can be done. This is one of my experience realizing the potentiality of barcoding. You see, I used to work with a zoologist, marine biologist. One of the marine biologists was working with this polychaete species. We're very confident that the specimen D and G are two different species because their key morphological feature is completely different. Okay, but when we did the barcoding, we compared both the genome. They're exactly same, 100% similarity. Not even single base pair is varying. So this made us to conclude they may not be a two different species. So when we examined very closely, we discovered this species is actually sampled from shallow regions of a coastal region, and this is from OMZ, that is oxygen minimum zone. Because the oxygen concentration is very minimal in those deep waters, they have developed a morphological adaptation. They have increased the cilia surface morphology to absorb more oxygen. That's why this appendages like growth, which is lacking here. Here they do have cilia in microfilament like form. You see, that's why the morphological adaptation actually enhanced their survival in that particular ecosystem. But for many classical taxonomies, they will instantly recognize them as two different species, which they are not. They are actually morphologically adapted. So having said this, I hope till here you guys have some idea about how the barcoding works. So what is this meta barcoding? Even a community of samples can be taken here, insects, worms, and I can extract DNA from the entire community. I can decode individual species because now the reference library is ready. We can actually compare even in the mixed population. We can extract the entire population DNA in one go. In PCR, you can select what phyla you need to study. Using the primer, you just you say, for example, I want only polychaetes from here. In PCR, I will use only polychaete primer to do the amplification of COI gene of polychaete. Then I can study the polychaete. Or if I want to entire diversity, I can sequence. I simply use a common metazoan primer, which will cover the entire animal biodiversity. Using Illumina platforms, I can have a individual sequences because we have built a complete database. Now we can decode the diversity. So what is the difference between barcoding and meta-barcoding? Barcoding uses single species for identification or for sequence synthesis. In meta-barcoding, community samples can be used. Even mixed samples can be used in one go. Okay, even these days, this is a proof. Even many plant food items preserved in the cans that consists of various plant species in one block. Actually, you can extract DNA from the block, from the fruit samples, and you can decode what all the species they have preserved in the food cans. Very much possible because we have created a database around DNA barcoding. So, having clear understanding of DNA barcoding and meta barcoding, I hope I did some justice. So I'm moving into eDNA barcoding, which is the seminar topic of today. Environmental DNA barcoding. That's what eDNA barcoding means. You don't need to sample the actual organism to study their taxonomy. You can simply sample something from their environment and decode their organism's diversity. Yes, you can simply collect 1 liter of seawater to decode entire fish population, bacterial population, plankton, zooplankton, phytoplankton. Everything can be decoded from just 1 liter of water. Okay, you are not collecting actual organism. You are collecting the media. Water or sediment or air can be collected to decode the diversity. That's why this is a non-invasive sampling technique. That's why we call them as an environmental barcoding. So how does this work? Okay, let's see one by one. So here is a proof. Just to detect the plant diversity of Amazon, they simply collected the air sample using drones and they have decoded the entire forest diversity. And in another paper, they compare the mechanism of the efficiency of eDNA barcoding. This row is this number actually indicates the number of species they recovered through the environmental DNA from the air. This is a traditional survey that has documented these species. It's very clear that environmental survey is very eDNA barcoding actually documents more species in the ecosystem than the traditional. Okay, imagine a non-accessible ecosystem like deep sea ocean or a deep cave region where you want to study a bat's population. You cannot go. I know people are going, but even when you go inside, there are a lot of crevices you cannot reach. You never don't know what is living inside. Okay, so in this work, they have a plot. You see, they installed some devices, something like this. Under this end, they kept the filter paper. You know, the filter for filtering the air that is passing through here. Actually, that filter paper will collect or bind all the free DNA particles that is carrying DNA. Everything is filtered here. So DNA extraction can happen from the filter paper. In the PCR, they will use a plant barcode primer to amplify the genes, and they will go for sequencing, compare it with the database. You know the plant species name. Okay. Actually, we did a small work, something like this, where we actually studied seaweed diversity of India, southern part of India, using barcoding. When we discovered that, when we went for eDNA barcoding, one of the problem we had is when we refer the eDNA sequences to the database, there is not much match. Meaning, the local species need to be barcoded first and created an efficient database. Then we realized we need to do the individual barcoding. So we collected more than 16 species, something like this, and we barcoded. These are the accession number of those species. And this is an ideal character of a gene. If the gene really has a variation which is relatively smaller within the species when compared to between species, you will see the red algae, they are all cladded here, green algae here, and brown algae here. Okay, it's a very beautiful phylogram, clearly showing that RBCL works on seaweeds. Even these days, when you're preparing a herbal formulations, the company is supposed to collect the air or the DNA that is in the air from the herbal and preserve the tissue paper for every batch of preparation. They need to preserve the tissue paper because from the tissue paper, we can recover the DNA to check the herbal, what are the herbs you have added for preparing the formulation. It is very much possible. So eDNA barcoding is not something very new. They are actually in practice in various business slots. See, those filter paper collected actually are DNA was extracted, and this is a gel picture of the PCR amplicon from where they have created the sequences and they found these are the herbs. involved in the herbal formulation. Okay. So it is very evident these days, just with 1 liter of seawater, you can decode from microbes to mammals living in an aquatic ecosystem, just if you have 1 liter. So this is how this is happening is, possible is actually the organisms are shedding DNA. Doesn't matter plants or animal, whenever they grow, they shed their DNA. Okay. So for eDNA barcoding, environmental DNA barcoding to work better, you see there is an interesting paper here. They use the entire global ocean system and landmass to collect samples to study their eDNA barcoding. But you can see this yellow indicates total biodiversity. Green dots are actually domain-wise diversity, and blues are kingdom-wise, and phylum-wise, a targeted species are in black dots. Okay, so what's been happening here is, um, they cannot go below kingdom. Okay, order, class, family, it's not possible because it's not getting matched. Okay, so the why it is not getting matched with the reference database, because the local population, they need to do the barcoding first in order to facilitate the better working of eDNA barcoding. Okay, so that needs to happen, but still there are many proof that people are collecting air samples to decode a particular diversity of a ecosystem. Even animal feces can be collected to decode the entire dietary composition, whatever plants they have feasted on. You can use ITS gene to check for a particular fungal presence. Okay, so the eDNA barcoding is finding in various expressions. So I just quickly want to show you that plant, animal, or whatever is happening in the aquatic ecosystem, their free DNA is actually released in the water. You can collect the sample, something like this, filter it, and from the filter paper, all the bound DNA, you can actually recover it, isolate it. You can go for PCR sequencing, and you can make a taxonomic database out of it. Okay, so once that database is ready, even any ecosystems samples can be collected, and we can decode it. For example, if you want to decode the biodiversity of mangrove ecosystem, I will collect the free water sample that has a free DNA. So there are filters, something like this, column filters. Through this nozzle, I will inject my water sample, I will pass it on here. All the free DNA particulates will be bounding to the column. So when I pass my 1 liter of seawater through here, I will seal one end and I will pass my lysis buffer that will facilitate DNA isolation. Inside the column, I will seal it, I will take it to the lab. So while I'm transporting it, every DNA is dissolved in the lysis buffer. I will just take the buffer in a tube. I will go for DNA precipitation, use the DNA for PCR, or even the entire genome. I can break it, Illumina sequence it. Now, all the sequences, I will find a match I will subject them to reference database. I will find a match, and that's how it works. Okay, simple. With 50 ml or 100 ml water, the entire ecosystem can be barcoded, because when an animal grows, they used to shed their DNA in the feces. Epithelial cells are peeling off, so the free DNA are proven to exist in water for 48 hours in case of schools of fishes. Even when the fishes are left, after 2 days, if you sample, their free DNA are bound to happen in the water, resisting autolysis, photolysis, enzymatic lysis of bacteria. They can resist for 48 hours. So you can even decode the history of an ecosystem. Who has visited? If you want to find whether this ecosystem have a particular invasive species or not, you can just have one primer designed to specifically detect that species. If you are getting PCR amplification in the across, you have an invasive species. If you want to know what species, you can sequence and you can know it. That's the power of eDNA barcoding. Not only water, you can use a fecal samples, air samples, soil samples. Many things can be used. Even there is an interesting study where a 50-meter core samples actually decoded, said that the 50-meter core sediment sample have a bacteria which are aerobes, meaning those actually are fossilized bacteria. The shore region actually are when deposition happens, they even able to decode the age of the sediment. Okay, and what era they may have buried, something like that. Okay, so eDNA can be done with air, water, soil, and many things. So how this works? If I want to study, say, for example, only a particular plant species from Amazon forest, I want to know how much diverse they are. What I will do, I will collect the entire air sample using drones. Those filter paper which is used to pass through the air sample will be used for DNA extraction. I will use only plant specific primer, RBCL primers, to get an amplicon. After isolating DNA from the filter paper, I will get one band, a high molecular weight band, and amplicon, I will get one single band. I will subject the band for sequencing. I will know the whether the plant is present or not. Or I can simply amplify the entire plant diversity, which will be of 10 million or 20 million reads using next-gen platforms like Illumina, HiSec, MySec, even NovaSec for longer reads. We can amplify the entire thing, and we know it will be up to 2 GB, 3 GB data per sample, and we can subject them to analysis, and we can decode the entire diversity. Usually plants, there is something called transpiration, where their water from the surface of the leaf will find the air. They are suspended in the air. While doing so, free DNA are actually released from the leaf surface because epithelial cells are continuously shedding. They are growing. They are subject to shed. The epithelial cells will have free DNA. Those mist can be collected through drones, suction pipes attaching to that through the filter paper. Those filter paper can be used to recover the DNA out. Okay, this is called non-invasive sampling. Even people are sampling coral reef ecosystems where you are not supposed to handle the coral reef with a proper permission. You can simply sample it now. A water sample will do. Okay, so people who are working with Sanger sequencing knows this. When you have individual sequences, you can go for nucleotide blast. But when you have a data of 2 GB, 3 GB of volume, there is something reference files released by ribosomal databases and even NCBI. You can merge your file with those reference file, and you can take out the taxonomy completely in an Excel sheet. Okay, so there are various applications of eDNA. Even migratory pattern of birds can be tracked from the fecal samples recovered from the trees. What species have visited, whose nest is this? Precisely to the species level, you can say population behavior, conservation, bioinvasion. Even when a ship is, ballast waters, when a ship from US come to our shore and releases water. If you want to know they are carrying an invasive species or anything, just before release, scan the water using eDNA barcoding. You will know. You can take a decision whether to let the water released in the Indian waters or not. Even people are using underwater drones to document the diversity. Okay, these drones, while traveling underwater, can collect the sample, extract DNA. They are, bring the samples back. Even I feel near future we may also have a compact sequencer inside this drone where we can get the live data in the lab when the drones are navigating under the water. Okay. For eDNA, you see, there are a lot of innovative application. People who are studying marine mammals knows this. They used to go to cruises, using binoculars. They used to watch the waters very carefully for the whale blow. Okay, these blown samples actually they will use to take a good picture of the pattern of the blow, because for blue whale there is a one pattern, and for sperm whale the pattern is completely different. So based on the pattern, you can decode the snout morphology, and then you will know what species it is. But still, you cannot sample your whale unless there is a stranding in the shore. You cannot go and take a whale's meat or something to, even for a good cost like assessing the health of the whale. But now using eDNA barcoding, it is very much possible. People are sending drones to collect the whale blow. You can time the blow when you're observing them from the distance. Send the drone sample. Drone samples are actually collecting in the filters, and from the filter paper you can recover, using the sea surface water as a negative control, or whatever diversity of microorganism are documenting from surface water, you can neglect it. Subtracting that, you can see the how the blow waters are unique in microbial diversity, or the blow waters actually telling us the health situation of the whale. Not only the microbial diversity in such a water can be decoded, even whale species, because their epithelial cells are actually thrown out, and you can simply recover the DNA. Use whale specific primer in the PCR, and you will get an amplicon to know the whale species. Okay, so it's not just with the whales. If you want to study a muscular diversity in the Amazon forest, not want to, it is already happening. This is a proof that people are sending drones to collect the air sample over the surface of the tree cover, green cover, and the eDNA samples are actually taken out from the filter paper. Then their entire diversity can be very easily decoded. So this is happening for coastal lagoon ecosystem monitoring to bioinvasion and many applications. Okay. So basically, you want to know eDNA barcoding is upcoming technology, and if PG students who are aspiring to have a long career in DNA sequencing, I would advise you to initiate your research in eDNA barcoding. I think this Barcode Biotechnology is most affordable say, DNA sequencing service in India. Such internship programs not only get you a sample sequence, but also will train you how to analyze the sequence and write some contribution in that area. Another beautiful thing about this company I like most is they have something called 1,000 Barcode Initiative, where they are encouraging people with discounts and many other things to barcode first yet to be barcoded species. For eDNA technology to work better, we need to have an enhanced local library. All your species barcodes should be present in the library so that eDNA can find its expression. Okay, so this 1,000 Barcode initiative is a very good initiative. You can be a part of it. You can send your samples. They will tell you, or they will also teaching. There are very videos to teach you how to decide a particular species is barcoded or not. How to use the species name to check in the NCBI. So that can be done. And the reason I'm again encouraging you to take DNA sequencing as a carrier is see the market is already 12.5 billion in 29, and it is going to project at least 16.5% growth every annum. Okay, and in India, the market is growing much more faster. You will see the sequencing market worth around, it is projected to work around $40 million by 2032. And it is growing at the enormous pace of 26.7% per annum in India. Okay, so I would say keep this DNA sequencing analysis in your radar. It may fetch you very good career in near future. And Barcode Biotechnology, thank you for them for inviting me to do this eDNA barcoding. And this 1,000 Barcode initiative is very much essential and very unique in business environment because you see this is a paper came this year which clearly shows the size of the bubble indicates number of barcodes produced by a country. Ironically, a country like India with rich biodiversity, we lack in cutting edge technology, and countries with winter frozen area like Canada, they have severe compared to India, their biodiversity is at least 15 times lesser, and they are producing more barcodes. Okay, they are actively. It's understandable. The University of Gulf is here, from where the concept of DNA barcoding has been expressing everywhere. So it is very much essential to create a first-time barcode, and people who are willing to publish a paper, I find it very tricky. If you have a species one sequence barcoded for first time, you can easily publish it. I'm not just dragging. I have done it. That's why. See, yesterday, a few days back, I just spent some time in understanding the top 20 endemic plant species. So this is how I used to find whether a biological species is barcoded or yet to be barcoded. How to find it? I took the endemic species because I thought many of them outside our country cannot access this. So I took the list of the species. When I watch it very carefully, all these red color marked species do not have barcode in NCBI. I see, for example, I took the Sophora interrupter. I just gave it an NCBI. There is no sequence. So if you can sequence it, similarly for animal species, these are endemic animal of India. All those red listed, the pictures are shown here. Those barcodes are not found in NCBI. So I just go to NCBI, select nucleotide, put the animal species, no data. Even if there is some data is there, no C1 sequence because C1 sequence is a barcode gene for these animal species. If they don't have C1 sequence, we are not considering this as a barcoding. So these species, if you can take easily, you can publish. Okay, this is a proof. Okay, I am not a zoologist, and when I came to this giant isopope, when I did the blast analysis, they had more than 5% variation with the maximum identity we can get from blast analysis. Then I discovered it is 5% variation means that species Kinsley and a D Disum spinosus is not there in the database. So our title is first barcode of Bathynomus Kinsley. See, we spent just 2 weeks in collecting the sample and 2 weeks for analysis. We took 1 week in writing the paper. In next 2 weeks time, it got accepted. Okay, it is around now 2 point something impact it is. So all these first-time barcodes are not only good for publication. PhD scholars and MSc students who are aspiring to have a publication, to want to go to European country to start their career, or BSc students who want to have one publication so that their international fellowships are considered positively by various European institutes. I think they should go for such a immediate work, and Barcode Biotechnology is actually facilitating this. They are encouraging barcoding of first-time species, and also they are teaching you through various free online videos right now available with this channel. You can use it to analyze your sequences and know how to get it. Okay. So this internship actually planned to give you two sample sequencing, free sequencing, using your own data. You can choose, even if you are a zoologist, botanist, you want to study your plant microorganism or animal microorganism, you can use these services. Simply, register for this internship. You can easily, first final year UG or second final year PG students, even PhD scholars, faculties, or anyone, even PUC. I see many teachers who are interested in research in PUC biology. They need to find how to publish it. Even we are basically from Bangalore, this Barcode Biotechnology. We see even school students are discovering new species. Last month, we had one student discovering a new species with the planaria using NCBS help. They have published a paper also. So school students and teachers are doing very good research job. They can avail this 21 days intense online bioinformatics training with their own samples. They send to Core Biotech and get it sequenced. They can use it for this internship. They may end up in a manuscript before 21 days. So that's all about eDNA barcoding. If you have any questions, don't hesitate to leave a comment in the video, and I will also be circulating your certificate form immediately after this. If you have a query, you can also use the form to ask us a query. Okay. So thanks for joining. That's all for now. If you have any queries, you feel free to visit the website or follow us in the social media platform. You can put your doubts there. We will attend to all your queries. Thank you for joining today.